entire skeletal muscle. Each muscle, surrounded by a connective tissue called epimysium, is a bundle of fascicles. Each fascicle, surrounded by a connective tissue called perimysium, is a bundle of myofibers. Each myofiber, surrounded by a connective tissue called endomysium and sarcolemma, is a bundle of myofibrils. Each myofibril has a structure of thick and thin filaments. The myofibrils give the muscle its striation appearance. The action potential arrives at the synaptic knob. The knob fills with calcium, forcing the synaptic vesicles, which are filled with acetylcholine, to merge with the cell membrane. Through exocytosis, acetylcholine is released from the membrane into the synaptic gap, where it is then picked up by acetylcholine receptors on the motor end plate of the muscle. Sodium and potassium channels open, allowing sodium to enter the muscle fiber and potassium to exit into the synaptic gap. The enzyme acetylcholinesterase enters the synaptic gap, breaking down the acetylcholine into acetic acid and choline. The positively charged sodium ions trigger a muscle action potential which travels along the sarcolemma and into the T-tubules. The terminal cisterna found on either side of the T-tubules of the sarcoplasmic reticulum hold a protein called calsequestrin, which binds to calcium ions and releases it into the cytosol. Calcium floods over the thick and thin filaments of the sarcomere. In order to fully understand the contraction of a muscle, we first need to look at the structure of the sarcomere, the basic building block of striated muscle. The most basic functioning units are the proteins myosin and actin, which form the thin and thick filaments of every sarcomere. Every thick filament consists of about 300 myosin filaments. The thin filament, actin, is accompanied by two other regulatory proteins called troponin and tropomyosin. Sarcomeres are separated from one another by a protein called Z-disc, which extends from one Z-disc to the next. In the middle of the sarcomere is the A-band, which extends the entire length of the thick filament. Is a zone of overlap, where the thick and thin filaments lie side by side. Just the opposite, the I-band extends the length of the thin filament and centers at the Z-disc. The H-zone, also in the center, does not contain any overlap, only thick filament. The M-line, appropriately enough, runs down the middle of the sarcomere. This M-line contains proteins that hold thick filaments together at the center of the sarcomere. With this information, we can understand how the contraction of the sliding filament mechanism works. Once in the cytosol, calcium binds to troponin, causing a change in the shape. The shape change moves the tropomyosin away from the myosin binding sites on the actin. The myosin heads attach to the binding sites, creating cross bridges. Myosin cross bridges rotate toward the center of the sarcomere, releasing ADP energy called the power stroke. Mitochondria releases ATP, which binds to the myosin head, causing it to detach from the actin. Once again, the ATP is converted into ADP. The calcium is then pumped back into the sarcoplasmic reticulum and attaches to the calsequestrin, allowing the tropomyosin to return to its resting place on the actin binding sites, resulting in relaxation of the muscle.